induced beta cell death model. Type 2 here is the DBDB model. You can see that just one injection can lower glucose to more than a week. So that's very effective. And this is also supported by the glucagon receptor knockout animals, where they present mild hypoglycemia and much lower insulin demand or increased insulin sensitivity. And there's also increase in alpha to beta cell transdifferentiation in this model, suggesting that may be a very good model. However, this has not been very successful therapeutic so far because it has some side effects. For example, a very prominent phenotype in the glucagon receptor knockout animals is an increase in alpha cell proliferation. And this is taken the paper from Maureen Sharon's group published about 20 years ago, showing that there is about 30-fold increase in alpha cell in the glucagon receptor knockout animals. In addition, and this can develop into glucagonoma, and this is also true in human as well. In addition to glucagonoma, the other side effect of anti-glucagon receptor treatment is dyslipidemia, which has been studied extensively by Snavasek, Dan Drucker, and others. And in addition to, there are also other genetic model indicating that it's the glucagon receptor in the liver that's responsible for both of those side effects. But to really move this those treatment to a clinic, we need to have ways to mitigate those side effects. For that, we need to understand the mechanisms of this glucagon signaling deficiency-induced alpha cell proliferation, which currently, which was unknown. So those two questions motivated us to try to develop a zebrafish model to study those added biology, to study specific those problems. And the reason we choose zebrafish is because it's a vertebrate, so a lot of physiology is conserved. And it's very easy to do genetic and chemical manipulation in this model. And that because the fish are somewhat transparent and small, so we can do live imaging in the model. In addition, in the early stage, there is only one islet in the zebrafish. So it's very suitable for studying the beta cell and alpha cell number regulation. So just showing that the size of the fish we start usually 1 to 2 weeks of age, those are about 3 to 5 millimeters in length. So you can put multiple fish in one well in a 96 well plate to do drug screens. And we can do genetic analysis very quickly by using CRISPR-Cas9. This shows some example we published when CRISPR-Cas9 was first described. In zebrafish, we show that by injecting into one cell stage of CADA and Cas9, we can have phenotypes in the injected fish right away. For example, if we look at pigmentation, that we can see 2 days later, we can create albino zebrafish. And these are stable. And in addition, we can inject multiple RNA to assess multiple gene function and study the interaction. Just as a sample, we choose tyrosinase and another gene, the DDX19, which is a nuclear RNA binding protein that are known when mutated to create a CNN necrosis phenotype. So in the fish that we inject both RNA, we see both phenotypes. So another easy thing to do in zebrafish is to generate transgenic animals. And this has been very efficient. And I'm going to show you here is a transgenic animal that expresses a GLP in alpha cells driven by the glucagon receptor, and expressing a H2B and cherry driven by the insulin receptor. And that we choose the nuclear localization because that beta cells are very compact in the core of the islet, just as in mice. And so it's very difficult to count if they're cytoplasmic. And we have validated those models indicating that every GFP positive cells are also glucagon positive, and every MTA positive cells are insulin positive. So as I mentioned, that we can also do imaging in zebrafish, and this is just showing an example showing that you can see beta cell proliferation. You can saw this too. Let me see if I can play it again. Okay, I mean, okay, it doesn't allow me to do that. Okay, so I will just move on. So with those, I'm going to tell you 2 lines of stories. I will just use alphabetical order, talk about alpha cells first, and then talk about beta cells project. So, the glucagon receptor is duplicated in zebrafish. So to really know if we can use zebrafish as a model to study this alpha cell proliferation phenotype, we need to develop a model, which is kind of risky because we have to knock out 2 receptors. This is a pre-CRISPR time, and knocking out is not that easy. And in addition, we do not know if even if we knock them out, if the beta cell, alpha cells are going to just behave like in mice or in human. That was unknown. But Ming-Yu Ni, when he was a postdoc in the lab, took this challenge and briefly generated the double knockout animals. And satisfactorily, he found that indeed in zebrafish, that the alpha cell also hyperproliferate in the double knockout animals. And this can be detected as early as 5 days of age. And this increase is primarily due to increased proliferation that as indicated by the EDU labeling. So you are going to see in the first part of the talk, we're going to show a lot of graphs about alpha cell number and also the EDU positive alpha cells. So those are our main assays. So with this model in hand, we collaborated with Alf Powers Lab, who is also interested in this problem. And we found the mechanism of this glucagon receptor deficiency mediated alpha cell proliferation is due to increased amino acid in the blood. And this is primarily, we know that amoeba are involved in amino acid catabolism to do glucose neogenesis and also for the urea cycle. And when this depends on glucagon, when the glucagon receptor is knocked out or disrupted, and the amino acids then accumulate in the blood. And for reasons we are still trying to understand that how the pancreatic alpha cells are very sensitive to the blood amino acids. And this amino acid one is to activation of mTOR presumably through amino acid transporters. Activation of mTOR then induces another amino acid transporter, glutamine, a glutamine transporter SLC38A5, and this positive feedback drives both alpha cell proliferation and the glucagon secretion. So using in vitro, we also found that the glutamine and arginine are the primary driver for this alpha cell proliferation phenomenon. I want to point out, though, that SLC38A5 is not expressed in the alpha cells at basal level, so it is induced by amino acids. In addition, knockout SLC38A5 only partially suppress the alpha cell proliferation. So those all suggestion that other genes are involved in this process. So what are those? So we took some candidate approaches thinking that if the beta cells are already more sensitive to amino acid, and if we know that mTOR activation is required for alpha cell proliferation, then we reason that maybe there is amino acid transporters that are highly preferentially expressed in alpha cells, and those may be involved in the alpha cells' sensitivity to amino acid and also immediate in the amino acid response. So we look into the database, on the safe database, we identified 3 amino acid transporters that are highly preferentially expressed in the alpha cells. For example, SLC7A2, which is an arginine transporter that are highly expressed in alpha cells compared to beta cells in both human and also in mice. And SLC38A4 is also enriched in alpha cells compared to beta cells. And SLC7A8 is expressed in alpha cells and beta cells. So taking advantage of the fast turnaround in zebrafish, we knocked out those SLCs. We found that indeed all 3 are knockout of all 3 can suppress the alpha cell hypoplasia phenotype in the double glucan receptor knockout animals. So now we have increased the number of genes that we know are involved in this alpha cell hypoplasia phenotype. That in addition to SLC38A5, we found SLC7A2, SLC38A4, and SLC7A8, which is an exchanger. So, however, all those transporters are thought to mediate mTOR activation. Then the question, we also now have confirmed in mice that SLC7A2 is involved, is necessary for alpha cell proliferation in both knockout glucan receptor knockout animals and also in antibody-induced alpha cell hypoplasia. And those work are going to in preparation. So the question we had is mTOR the whole thing, or have we identified the entire pathway? So to answer that question, so we asked a simple question, that is activation of mTOR1 sufficient to induce alpha cell proliferation? So to answer that, Yulong Gong from a CS6 sponsored a graduate student in my lab for a year, took this challenge. And so first he generated transgenic zebrafish that express a constitutionally active mTOR or a constitutionally active RAP, which is upstream activator of mTOR. In those animals, he did not see an increase in alpha cell number or alpha cell proliferation, but he indeed see an increase in alpha cell size, suggesting that activation of mTOR alone is insufficient to cause alpha cell proliferation. However, we know in our in vitro model that hypoaminoacidemia are both necessary and sufficient to cause alpha cell proliferation. Then he said that he proposed a model where maybe the amino acid not only activates the mTOR, but also activates other signaling pathways, that they collaborate together to induce alpha cell proliferation. So he looked into the literature and found that there is this class of GPCRs, this is class C GPCR, which usually have a very large N-terminal extracellular domain that are sensitive to amino acids. We are not interested in those glutamate metabolic, the glutamate receptors, because we know that glutamate is not involved. And so we focus on 3 GPCRs, CASR, GPRC6, and also the taste receptors. And we knocked them out. We found that the other 2 does not decrease alpha cell hypoplasia phenotype in the double knockout animals, except for CASR. So indicating that CASR may be participating in this process. So what is CASR? So CASR is the calcium syncytin receptor. And it's discovered because its role in in extracellular blood calcium modulation and several diseases related to that. And in addition, it also has non-calcitropic roles in addition, such as in insulin and glucagon secretion, and also in nutrient sensing in the gut. And it is usually form a dimer on the plasma membrane and a signal through the GQ, GI, and beta arrestin. And it can be positively allosterically modulated by most of amino acids. So to see if amino acid is indeed involved, we made triple knockout animals and suggesting that in the triple knockout animals, proliferation, alpha cell proliferation is brought down to the minimum level, just as in wild type. In addition, this is also true in mice in the ex vivo mouse culture model using the Cas inhibitor CalHex231. We can almost completely block the alpha cell proliferation induced by high amino acid. So this suggests that Cas may be involved. And then, so this is Cas necessary, or is the Cas acting in alpha cells? To address that, we generated a transgenic model expressing human Cas specific in alpha cells. And we found that by rescuing Cas expression in alpha cells in the triple knockout animals, we can rescue both alpha cell proliferation and also restore or increase the alpha cell number, suggesting that Cas is acting in the alpha cells to mediate this alpha cell proliferation response. So I mentioned that Cas acts through multiple signaling pathways. We would like to understand which signaling pathways is involved in the alpha cell proliferation induced by amino acids. So to address that, we use a DRAD approach. For those who do not know, DRAD is just a modified GPCRs that do no longer responsive to the endogenous ligand, but responds to a synthetic, very stable ligand. In this case, in most cases, the CNO, the clozapine and oxide. So most of the drugs are modified from either human, rat, or mouse muscarinic receptors because they have multiple subtypes coupled to different signaling pathways. So we express this GQ and the GQ and GI coupled drug in the alpha cells and try to see which one would help alpha cell proliferation in the triple knockout animals. So we found that GQ, but not GI activation can restore alpha cell proliferation in the triple knockout animals, suggesting that the Cas likely mediate its proliferative response through the GQ signaling pathway. So just showing here that in the triple knockout with the transgenic expression of GQ drug in alpha cells, in the absence of the ligand CNO, the beta cell number is just very low to the basal. But in the presence of CNO, which stimulate the GQ, then we can increase the alpha cell. So in addition, we ask the question if we can activation of GQ and mTOR together in the GCGR intact animal can also cause increased alpha cell number and also alpha cell proliferation. And indeed, the data shows yes. So you don't have to pay attention to all the other controls. Just pay attention to here that in the triple knockout and in the double transgenic animal that has constitutional activation of RAB or therefore mTOR1, and also expression of the the GQ dread. And in the absence of the ligand, the alpha cell number does not change. Those consistent that with our previous data that activation number 12 alone is insufficient to cause alpha cell proliferation. However, if you also active GQ in this model, then alpha cell number increase, alpha cell proliferation increase. So suggestion that co-activation of mTORC1 and the GQ pathway are sufficient to induce alpha cell proliferation, even in the absence of glucagon receptor knockout or hyperamyloidosis. So we know that in GQ-mediated alpha cell proliferation is usually through the ERK kinase pathway. So we asked the question if that's also the case in alpha cell proliferation. What we found is that indeed that ERK activation, as indicated by phospho-ERK immunostaining, is coincide with conditions that increases alpha cell proliferation, and inhibition of ERK1/2 using a MEK inhibitor can suppress alpha cell proliferation in both the double knockout animals and in several other models we have that can increase alpha cell proliferation. In addition, there's inhibition of MEK can also reduce or suppress alpha cell proliferation in the ex vivo cultured mouse islet induced by high amino acids. So to quickly summarize what I have told you, that is, we think that amino acid acts through at least two pathways, either both the SLC-mediated activation mTOR and also the CADA-mediated GQ pathway, that they collaborate to cause alpha cell proliferation. And there are also other interesting observations we found in this project that I'm not going to go very deep into, suggesting there's a crosstalk between these two pathways. So this study then identified a novel function for KSR in amino acid sensitivity and also amino acid mediated alpha cell proliferation. And now I'm going to move on to the next topic on beta cells. And so this is our first attempt trying to model diabetes in zebrafish, especially type 2 related diabetes. And this was done by my first postdoc, Nizat Madison. And she tried to test if zebrafish have the compensation and decomposition. And she used both nutrient approach and also diet approach and also genetic approach. And first she tried using nutrient approach. So she was finding ways to try to make fish to become fat faster. And she found a very convenient way, that is, she found that fish seems to like chicken egg yolk. So by culture them into a 5% egg yolk solution, she found that by 8 hours, she saw a very significant increase in beta cells. However, when she tried to do that multiple days, trying to induce decompensation, she failed because the fish died before the beta cell dies. So it was somewhat disappointed, but Min-Yu, when he joined the lab, again, said that this is interesting, I want to know why this happens. So just a very quick summary of what he has found is that they found that this is due to a persistent activation of the nutrient secretion component pathway, and that induces ER stress. And for reasons we still do not know, this ER stress seems to trigger the release of FGF1, and FGF1 induces differentiation of the progenitor cells to become and to get more beta cells to maintain glucose homeostasis. And so going back to try to model this decomposition, so since the nutrient approach did not work, so Nizet tried a genetic approach, mimicking what has been described by Derek Narosa's group by expressing a dominant active IGF-1 in the muscle. And she found that indeed there is type 2 diabetes development, also beta cell compensation and decompensation. However, this takes a long time. So that's also not very ideal in the zebrafish because we want to see things happen faster so that we can understand the mechanism. So then she thought that maybe we can combine both approach, but before she can finish the project, she was offered a very nice position at Washington State University. So she just took that position, and I had to wait a couple years to get Bingyuan, which I'm going to talk about just in a second. I'm ahead of myself. So she said, "Oh, why don't we just combine these two approach and just feed those muscle insulin resistance models, we call them ZMIR, with overnutrition due to multiple days." And doing that, she found that indeed she can mimic the compensation phase and decompensation phase in about 4 days. So in the first 3 days of overnutrition, after each phase, she saw a significant increase of beta cells. However, during the night prior to the fourth round of overnutrition, she saw a very significant decline of beta cells. And she looked at the time course more carefully and found that this beta cell decline occurs about in a 4-hour window. And as a result, that the fish become also diabetic. And I want to clarify here that we cannot measure blood glucose in a small zebrafish. What we are measuring is the total glucose, free glucose in the larva. We just grind the larva up at the total larva up, measure the glucose. And we think most of those are extracellular glucose because most of the intracellular glucose are phosphorylated, which cannot be measured using this method. And we also think that this increased glucose control may not due to beta cell death, because in other models, until you have more than 50% of beta cell loss, you do not see glucose homeostasis defect. So we think there is also beta cell dysfunction going on. And we will actually provide some data here later. I will get back to that. So this rapid decrease and also an absence of apoptosis markers, because we did a lot of imaging, we never seen any fragmented nuclei, so suggesting that maybe it's not, beta cell did not die through apoptosis. That is to propose that maybe the beta cells are killed by macrophage, because the macrophage are known to cause beta cell death, to kill live cells in other systems. And so she generates transgenic phage that express then marks both the macrophage using this MPEG-1 promoter and the beta cells, and did live imaging. And what she found is that indeed there is macrophage coming in only in the ZMR fish, not in the control animals. And in addition, the macrophage seems to wrap around beta cells. So she was convinced this will lead to beta cell death. And we'll just show you. So I just want to clarify that those are not resident macrophage, because in zebrafish, resident macrophage doesn't appear until 45 days later, 45 days of age, and this is about 9 to 10 days of age. So we do not have resident macrophage. So just showing you a quick movie about this neutrophils, macrophage. So you can see that there are macrophage never come close to the islet in the control animals. Whereas in the ZMR-fish, you can see macrophage approaching the islet and interact with beta cells, and then they also retract. So that brings me to just another detour on islet inflammation. So this, our data suggestion there is islet inflammation in our model. And islet inflammation is a common feature to type 2 diabetes. It has been reported in multiple studies, both in human and also in animal models, that there's an increase in macrophage. And you have probably also heard from Wenxian just last summer talking about islet inflammation. And the proposed model, most people think that is initiated from resident macrophages, but there is also evidence, mostly from in vitro, that when under stress condition, beta cells can also express inflammatory cytokine, which may also be responsible to initiate islet inflammation. However, it's unknown that if beta cell produced inflammatory cytokines are sufficient to induce inflammation, because they are very low in quantity. So because our model do not have resident macrophage, so we wonder how in this model islet inflammation is initiated. And we got some idea from a chemical analysis trying to identify how the beta cell is lost. We thought it's beta cell death. We used multiple drugs that inhibit different pathways of beta cell death. As you more know, that there are multiple ways a cell can die. That can be classified into apoptotic death or necrotic death. And the most commonly studied two pathways, the apoptosis and necroptosis. And apoptosis can be inhibited by ZVAT, which has been commonly used also in zebrafish. And necroptosis can be inhibited by NEC1, which inhibits RIPK1, and GSK, which inhibits RIPK3, and NSA, which inhibits the MLKL, which is the pore that is involved in forming the membrane rupture during necrosis. So we use those drugs, and we confirm that, as we just suspected, that ZVAT does not suppress the beta cell death we saw in our model. However, NEC1 and GSK can both suppress beta cell death, but NSA does not do it. So suggesting that this cell death we are dealing with is not a typical necroptosis or apoptosis. And to this day, we're still trying to figure out how those cells died. But that aside, so we just, we thought, okay, those are just the chemicals. We're not so sure if that's so clean. So we made knockout of RIPK1 and RIPK3. And indeed, in both knockout animals, the beta cell deaths do not occur, and the increased glucose does not happen. So suggesting that those two genes, I'm not showing the RIPK1 data here. So then we ask if the RIPK3 could act anywhere, is it required in beta cells? So I would say that generating a cell type specific knockout in zebrafish still have a lot of work. So we took the shortcut by expressing a dominant active of the kinestat RIPK3 in beta cells specifically. We show that it's expressed only in beta cells. And we also show that in the ZMR fish, the distribution of RIPK3 is very different. It forms punctate indicating activation. So supporting that in those beta cells, RIPK3 is activated. And in the dominant active expression animals, beta cell death is prevented and the glucose homeostasis is preserved, suggesting that RIPK3 is acting in the beta cells. The same is also true to RIPK1. So I'm just going to, because this has been published, I was just trying to give you some skip over a lot of experiments, just trying to tell you that we found that RIPK3 is activated by ER stress. And then we wonder how this RIPK3 causes inflammation. So we look at the inflammatory cytokines. And for example, we look at IL-1 beta, which is the most commonly associated with inflammatory cytokine involved in islet inflammation. We found indeed in the ZMR phage overnutrition induces IL-1 beta expression in the beta cells, and the expression of dominant active RIPK3 prevent that from happening. So suggesting that RIPK3 mediates IL-1 beta expression, and we have shown that's also true that in cultured mouse islet. So using palmitate as the ER stressor, we found that in the presence of GSK inhibitor, GSK871, the RIPK3 inhibitor, that IL-1 beta induction is suppressed, and that does not affect the ER stress markers. So in addition, we found that knockout RIPK3 or IL-1 beta prevented the islet inflammation as indicated by the macrophage infiltration in the islet. So these pathways are not only conserved in mouse, and it's also conserved in human. Okay. So we took advantage by collaborating with our powers. We took advantage of his system where he can transplant human islet into NSG DTR mice that are immunodeficient and also expressing this deficit toxin receptor in the beta cells. And he just transplant a suboptimal amount of islets so that once it kills the endogenous beta cells by injecting DT, the human islet alone cannot maintain glucose homeostasis, so that they are diabetic. And then this is a way to study the effect of diabetes or hypoglycemia on human islet. So we took some of the kidney sections and studied whether we can see RIPK3 activation, stress, activation of kappa B, and also the induction of IL-1 beta. And we found that indeed in those human islet cells, we do see an increase in ER stress activation of RIPK3 and activation of the P65, which is a subunit of kappa B, and also increase of macrophage infiltration in those transplant tissue. So suggesting that this access is conserved also in human beta cells. So this is just a graphic summary. Well, I just told you that overnutrition induces ER stress. I think I did not want to go too much in detail to, but we think is that it's not just a mild ER stress, but it has to be very intense ER stress that activates RIPK3 and RIPK1 and RIPK3 that through an kappa B pathway to induce IL-1 beta expression in the beta cells, which we do not know if it's direct or indirect, that needs to be recruitment of IL-1 of macrophage. But the question still remains that we lost beta cells. How does that happen? Is macrophage has anything to do with beta cell loss? So to answer that, we thought that if we can get rid of macrophage, if we protect beta cells, then there's also at least macrophage is necessary for beta cell mass. And the experiment, we did 3 types of experiment by collagenate liposomes, as everybody uses in mouse, and also we use it in zebrafish, and we also use the mutant that do not have that has macrophage deficiency, the IRF8 mutant, that has very few macrophages. And also in another model where we can ablate selectively at the macrophage using a prodrug mediated cell killing approach. So this approach takes advantage of bacterial nitrate reductase, which can convert a antibiotics metolidazole into a cytotoxic compound, which can then cause damage and cell death. And this has been shown to have very limited, very minimal bystander effect. So just kill whichever cell that expresses the NTR. So we express the NTR in macrophage fused to mTERI, as you can see here. Using metronidazole, we can kill substantially, the macrophage substantially. In all the three models, we can prevent the beta cell death and preserve glucose levels. So suggesting that macrophage are necessary for the island inflammation, for the beta cell loss, and also for the loss of glucose homeostasis. However, as you probably have noticed already, that in the movie that I showed you previously, that although we saw very close interaction between macrophage and the beta cells, and once macrophage left, the beta cell were still intact, suggesting that at least macrophage is not mediating beta cell death by phagocytosis. So maybe then it mediates by inflammation. So in zebrafish, that macrophage biology is still very rudimentary. So we do not have a lot of characterization of this pro-inflammatory versus anti-inflammatory cytokines. So in the field, we use TNF-alpha expression as a marker for pro-inflammatory cytokines. So then we look at if those macrophage in the islet or near the islet are pro-inflammatory. And as shown here, we show using in situ hybridization that indeed the macrophage as marked by this MTAG1 expression do express high levels of TNF-alpha. In addition, using a different approach, using a transgenic phage that reports the TNF-alpha expression is using this TNF-alpha promoter-driven EGFP line. We also show that macrophages that are nearby the islet, as shown here, we also macrophages marked as grabbed here as well, that they have higher expression of EGFP, but the macrophage farther away do not have EGFP, detectable EGFP expression, suggesting that somehow macrophage close to the islet either have higher TNF-alpha expression, either those macrophages are preferentially attracted to the islet or islet is inducing TNF-alpha expression. We cannot distinguish these two possibilities at this time. And if we knock out TNF-alpha, then we can rescue the beta cell death and prevent beta cell death, and also prevent the increase of glucose. And we think that the TNF-alpha works in macrophage because if we just rescue TNF-alpha expression in the macrophage in the knockout animals, we can recapitulate the wild type phenotype. That is, beta cell death still occurs and glucose still increase. So suggesting that it's the beta cell derived TNF-alpha that's responsible for the beta cell death. And then because neutrophils and beta cells usually come in tandem during inflammation, and we also look at a neutrophil, see if there is also neutrophil infiltration in the beta cells. And I'm going to show in the movie that indeed, we see a lot of neutrophil coming in and they move in very quickly into the islet. In addition, that we do not see any macrophage, any neutrophil coming in, or very few neutrophil coming to control animals. In addition, after this period, we do see that neutrophil usually come to one or two spots, and it's usually those spots that we see beta cell loss. So implicating that neutrophil may be involved in the beta cell loss. And to determine if neutrophil is necessary for the beta cell loss, we use two different approaches trying to manipulate the neutrophil function. In one, we expressed a dominant active rho A, which is necessary for neutrophil chemotaxis, or neutrophil move to the target site, and that can prevent beta cell death, but do not prevent the loss of glucose homeostasis. So now this become clear that beta cell loss and that the increased glucose is two separate phenotypes. So we also use the approach, the cell ablation approach I just mentioned to you, that we apply to macrophage to do the same thing to neutrophil. What we found is also that if we ablate neutrophil, we also prevent beta cell death, but not the hyperglycemia that we observed in the animals. Again, suggesting that the neutrophil is necessary for beta cell death, but not the beta cell dysfunction. So how did neutrophil get in? So with this, we look at the prime suspect, which is IL-8 or CXCL8, which is the most potent neutrophil chemokine. We look at CXCL8 expression, and we found that prior to a neutral macrophage coming in, there's a very little CXCL8 in the islet. But once macrophage comes in, and we see a significant increase in CXCL8 expression in the beta cells. And and we found that this induction requires TNF-alpha, and also the other genes that we mentioned already. Come on, Maris. Okay.
We are like
so so knockout of IL-8 or CXCR8 all suppress neutrophil infiltration, and so those data all suggest that it's the beta cell derived CXCL8 that's responsible, because we did rescue. When we rescued CXCL8 in beta cells, we can now induce a beta cell death. But if we rescue it in the macrophage, that does not rescue the beta cell death phenotype. So that's just a quick graphic summary that I told you that EL stress activate a RIPK3-mediated induction of IL-1 beta that leads to recruitment of macrophage. Then macrophage induces CXCL8 expression, which recruits neutrophil, and which then leads to beta cell loss. And how this happens, we still actively investigating. So those are the questions we're trying to address. So why only some beta cells lost, and so that's probably implies there's some heterogeneity there. And why beta cells lost during a specific time window? Is it circadian, or is just takes time to reach that point? And how does beta neutrophil cause beta cell death? And what is the physiological function of islet inflammation? Because this seems to be a conserved pathway. So with that, I just trying to acknowledge people and funding agencies and take your questions.
Thank you, Wenbiao. So there is a question in the chat. asking, do any of this occur in alpha cells?
No, this is very specific to beta cells. So when we look at alpha cells, alpha cell seems to be unbothered, and that the number does not change at all. And I think we also look at the glucagon expression, that doesn't change at all.
Thank you. Do we have any other question? Please just feel free to ask directly.
Wenbiao.
Hi, Mina.
Hi, great talk and beautiful work. Very impressive and unique models. Congratulations. Beautiful work.
Thanks.
Well, I have two small questions for you. So I think in the first part of your talk, you showed us 3 amino acid transporters are involved in the phenotype you observe, and I believe it's alpha cell hyperplasia, right? In that double knockout model. So you think that 3 transporters are responsible for that phenotype? Mm-hmm. I wonder whether these 3 transporters, amino acid transporters, are implicated in diabetes or not. I know you use the zebrafish model, but I wonder whether I'm curious about the implication of these 3 transporters.
That's a very good question. I have not looked into the GWAS studies, so that's a very good suggestion. I should have looked into those and see if they're involved in diabetes. I think SLC72 has been implicated in diabetes, but I do not know the other 2.
Okay. So another related question is you talk about the crosstalk, and actually you mentioned about the potential crosstalk between signaling and ERK in driven that phenomenon. But my question is, I think you mentioned the GQ, right?
Yes.
And your model is glucagon and glucagon receptor knockout. So I know PK pathway is very important in that scenario. So I wonder whether PK pathway is involved in your system.
Insulin, you said?
PKA signaling.
PKA. So because the CASR rarely, I think maybe in a couple of instances, papers that describe that CASR can activate PKA. So we did not look into the PKA aspect.
Okay. Thank you.
Thank you for the questions. Very good.
Great talk.
Thanks.
Wenbiao, I have a question. It's Inchin here.
Yeah.
Hi. So it's a beautiful talk. I learned a lot, and the
Thanks.
question I'm going to ask is just for my own knowledge. So when you say you deplete macrophage, right? So
yes.
When you say that, so actually macrophage and neutrophils, they're all from monocytes. So monocytes will differentiate into macrophage and neutrophils and others. When you say, so you have a specific depletion of macrophage, or when you deplete macrophage, actually also deplete neutrophils and other
So
things.
the promoters we use are more differentiated markers. So like MPEG-1 stands for macrophage expressed gene 1. And the neutrophil promoter we use the lysozyme promoter, which is only expressed in the neutrophil, I think.
Okay, thank you. I learned something.
Yeah, thank you.
Okay.
So Wenbiao, there are a few more questions in the chat.
Yes.
One is from Wei Ping Han. Question is, for the alpha cell proliferation studies, did you examine individual amino acids?
So yes, in the in vitro we did. So the conclusion is that you need a general high amino acid. So that means if you just use one amino acid, you're not going to induce alpha cell proliferation, no matter how high it is. So there has to be a high alpha amino acid tone in the environment. But so what we did is by dropping out individual amino acids, we cannot drop out lower the concentration of glutamine or lower the concentration of arginine. Other than that, you can lower any other amino acids individually.
Thank you. And another question from Xiaoqing Tang asking, is there any beta cell dedifferentiation occurs prior to beta cell death?
Right. So that's a very good question. We do not have any evidence for that. So we have looked at the insulin expression in all the cells prior to beta cell death, and every cell that M-cherry positive are still very strongly stained with insulin. So, but I cannot, maybe that's not the best marker, but that's the marker we have looked so far.
Thank you. And there's a question about the necroptosis, asking whether you try to perform the TEM to confirm the beta cell necroptosis.
Right. So first of all, we do not know if it's necroptosis yet because we cannot suppress it with the NSA, which is the last step that inhibit the executioner of necroptosis. So we are not certain that it's necroptosis. So we are still investigating what type of cell death that beta cell undergo. And once we know that, we will do TEM analysis, just trying to prove morphologically. Thank you for this question.
Yeah. So and there's another one along this line from Zhang Weihua, asking what is the exact cause of the ER stress in beta cells? So
it's we know that beta cells are very prone to ER stress because it has to make a lot of insulin. And making insulin is a difficult thing to do because you have to process them by cut them into pieces and also fold them. So it's a very intensive work for the beta cells. And once you make an animal insulin resistant, they have to work even harder. So that's the trigger of ER stress. And on top of that, we give them over nutrition that is making the ER stress even more severe.
Yes. And another question from Zhe Xian, asking, does glutamine also regulate beta cell proliferation and function? What is the source of glutamine in vivo?
Let me think. So I did not talk about this at all. So the source of glutamine is mostly from, I would say probably from muscle. So we know that alanine and glutamine are the primary amino acids in the bloodstream. Those are the two highest concentration amino acids in the bloodstream. So it's most likely from muscle, and it's also involved in the ammonia or the amino acid homeostasis. Not ammonia homeostasis or transamination in different tissues. So that's the source, I would say, is most likely from muscle, although it could be from diet. And in our model, we do see a mild increase of beta cell proliferation in the glucagon receptor knockout animals. And that's one of the paper I mentioned that's being submitted that we're going to discuss on that.
Okay, thank you so much.
Hi, Wenbiao.
Still up?
Hi, Wenbiao. Can you hear me?
Yes.
Hey, hi. Hey, very nice talk. I think I'm learn a lot from your talk. My question is, because the beta cell is quite heterogeneity is quite different from each other. I'm thinking about what kind of beta cell is more sensitivity to the macrophage or neutrophil. Is there any such as more mature or more little less mature beta cell is more sensitive, or there's any markers sensitivity to these immune cells?
Okay, very good question. And so we have looked at the lineage. So in beta cells, probably also like in mice as well. So beta cells are derived from two lineages, either dorsal bar derived or ventral bar derived. And in the dorsal bar derived cells, that forms earlier in zebrafish. And those cells are usually not as mature though, because they express lower concentration of GLUT2, GLUT1, and also lower concentrations, lower levels of insulin. So those are considered early derived but less functional beta cells. Whereas eventually derived beta cells develop later, but they're more functional. So we try to see if we can distinguish those if those cells are more sensitive or less sensitive. What we found is that there is no difference. So at least from that standpoint, we have not found what makes. We know that there's heterogeneity in beta cells, but we do not know what kind of heterogeneity make them more prone to death. It's most likely either more ER stress or more oxidative stress, but we do not have the definitive data yet to address the question.
Okay, thank you.
That's one of our ongoing struggle.
One more question is for the alpha cell project. Is the CASR, the CASR, because I think the CASR is, you mentioned the CASR is very important for the calcium sensing, right? Calcium release. Is there any effect or affect the glucagon secretion? Is there any method to measure the glucagon secretion? Or I think this may be very important for the alpha cell function.
Right. So good question. I think there was only one clinical report that in a CASR mutant individual that has hypercalcemia, that has also hypoglycemia, but I do not know if this has been commonly observed. So I want to emphasize here that we do not think that CASR really play a role in normal amino acid levels, but it's only when blood amino acid is increased did the CASR activity increase in beta cells, in alpha cells, and leading to alpha cell proliferation. So it's I would say maybe it's a somewhat pathophysiological phenotype of CASR.
Thank you.
A function of CASR.
Great. Do we have any other questions? If not, thank you so much, Wenbiao, for a wonderful talk. We all learned a lot. And thank everyone for being here. I'll see you next time.
Yeah, thank you everybody for being here. And thank you for all the good questions. And if you have any questions or suggestions, you can just WeChat me or email me. Thanks.
Bye-bye, everyone.
Bye.